Dear students, today's class topic is cancer biology and therapy updates. As we all know that when normal cell evades its normal cell regulatory pathways, then it will become cancerous. And there are a few characteristics of cancer cell, which includes evading cancer cell generally evades apoptosis, which is a normal cell death mechanism which regulate the normal cell growth. And when a normal cell complete its life cycle, then it would die via apoptosis. And any kind of inhibition or evasion in the process of apoptosis would lead to the cell into the cancerous formation. The other characteristics of cancer cell is uh, uh, it is self-sufficient in growth signals. That means for a normal cell, there is a requirement of few growth factors, which via binding on its growth factor receptor stimulate downstream signaling cascade, which regulates cellular growth and proliferation. If uh, if these signal will stop, then normal cell would die. Then normal cell would die. However, in case of cancerous cell, there is no outside requirement of growth signal, because cell become somehow self-sufficient in producing these signals and thereby maintaining its continuous growth. The another famous hallmark for cancerous formation is its anti-growth signals. Like in normal cell proliferation conditions, the anti-growth signals are provided to cell to stop its growth whenever there is alteration in the normal cellular physiological processes. If cell become insensitive to these anti-growth signals, then cell would continuously divide and grow in order to make its demand fulfilled, and thereby it changes in cancerous conditions. And there is one more important thing, that once the normal cell become cancerous, it become more invasive in terms of growth. It actually invade the surrounding neighboring tissues and slowly get metastasized to the other part of the body. Metastasis is an important process in cellular cancer growth and development. Cell grow uncontrollably, and slowly it get through invasion, it get entered into the neighboring tissue, or spread into, or we can say that spread into the neighboring tissue, or what other thing it would lead when there is excessive growth. The few cells from the tumorous part get detached and entered into the blood circulation, and via blood circulation they get spread into the whole body. And this is the process of basis this is the process of metastasis, which is the important hallmark of cancer cell. There is another important factor which is exhibited by cancer cell is limitless replicative potential. That is, its replicative machinery somehow get, due to various kind of mutations, it replicates its chromosome or its DNA or its genome, replicate uncontrollably and indefinitely, and this would lead to the excessive cancerous growth. The another hallmark of cancer is sustained angiogenesis. Here, as cancer cell grow uncontrollably, there is increased oxygen demand, and to fulfill these oxygen demand of the cells, cancer cell induces angiogenesis within within the tumorous growth. Angio means formation of new blood vessels, which could supply excess blood and oxygen, which is a requisite for the cancer cell growth and development. So these are the few hallmarks of cancer cells, and how normal cell become cancerous when uh, cell cycle control get uh, disturbed. So what is the mechanism behind the formation of a tumor? There is a three-step mechanism: the initiation, the promotion, and the progression. Actually, mechanism of carcinogenesis lies or started, initiated when a DNA get damaged or a DNA of a cell get mutated. Then due to this mutated, normal cell become malignant cell, and this malignant cell, once it get malignant, it started proliferating. As you can see with this diagram, the proliferation of malignant cell. And after it attains certain level of growth, it started angiogenesis, migration of cells from one side to another side of the body, and it invades surrounding tissues, and thereby it facilitate the process of metastasis. And these are the characteristics of cellular proliferation in the promotion state of cancer. And thereby, when all the characteristics of cancer cell will become uh, full potential in terms of its progression into the other body parts, it will lead to the cancer. There are actually, I already discussed about the hallmarks of cancer, and as you all are well aware of each characteristics, we can target each one of them for designing of therapeutic implications to treat or to cure cancer. Like as we start with a cyclin dependent kinase inhibitor to stop the evading growth to stop the growth suppressor, which is growth suppressor is an important hallmark of cancer cell. Here we can try to just use cyclin dependent kinase inhibitor, which can inhibit the growth by evading growth suppressor mechanism. We can also use immune activating anti-CTL, that is cytotoxic T lymphocyte A4 monoclonal antibodies, to prevent the avoiding of immune destruction. Cancer cell exhibit an important characteristics that it actually avoids the immune mediated self-destruction. If we can, if we introduce the anti-CTLA4 monoclonal antibody, there may be chances to kill the cancer cell by enhancing the immune mediated destruction. There is another hallmark of cancer cell, that is there is enabled replicative immortality. In terms of cellular proliferation, we can use telomerase inhibitors to inhibit the telomerase function, which is actually responsible for the immortality of these cells, and thereby we can suppress the tumor growth. The another characteristics of cancer cell which we can target is tumor promoting inflammation. In this, we can use selective anti-inflammatory drugs. Actually, inflammatory is known to induce the cancerous situations in many of the carcinogenesis. Here, anti-inflammatory drugs have potential to just stop the further tumor-led inflammation, and thereby they can inhibit the tumorous growth. And we can also use inhibitors of HGFC MAT. Actually, these inhibitors stop invasion and metastasis through cell cycle signaling pathway. Like we earlier discussed that angiogenesis is an important hallmark of cancer. We can use vesicular endothelial derived growth factors, VEGF inhibitors, to stop the angiogenesis, and thereby we can inhibit the cancerous growth. We can also use PARP inhibitors, which actually target the genome instability and mutation of cancer cell. If mutation and genome instability can be taken care of, there would be chances to suppress the cancerous cell growth. Another characteristics which we can exploit to develop a therapeutic target is resisting cell death. Actually, cancer cell resists cell death, and this happens because they actually produce pro-apoptotic BH3 mimetics. Actually, and they start antagonizing activity of various anti and pro-apoptotic factors. So we can use pro-apoptotic BH3 mimetics to target the cell death resistance. There is another factor which we can use to target is deregulation of cellular energetics. Actually, we can use here aerobic glycolysis inhibitor, because cancer cell overexpresses glycolytic enzymes to fulfill its energy needs. So we can use inhibitors of glycolytic enzyme, and thereby we can directly starve the cancer cell and thereby inhibit the tumorous growth. We can also use growth factor receptor inhibitors. Actually, as I earlier discussed, that growth factors are very much important to sustain the proliferative activity and via signaling cascade. If we use inhibitors such as in this case we used epidermal growth factor receptor inhibitor, we can target the cancer cell, and there is various researches going on in or under in various researchers under clinical trial, that by targeting EGFR, HER2, PDGFR receptors, we can suppress the tumorous growth. So this is for now. We will continue this topic in our next class. Thank you, students, for your kind attention.